So welcome. And I just want to give like a brief sort of introduction about why are we doing these bridging theories and experiments. And today, Vincent, who works on the human, he just told you the stem cells, and I, who's a theoretician, we seem to ask the same questions, but we're coming from a different perspective. And one thing we're trying to tackle, part of this virtual human development, is to be able to merge what we know from experiments and what is known in theory and try to see if we can fill the gaps or see how we can work together in order for us to make this virtual human in silico. And to do this, the first thing we have to do is be able to talk to each other. And this is part of this discussion here. And so I'm grateful for everybody that joined us. And I'm really grateful that Vincent agreed to give a talk at the same time. So the floor is yours, Vincent.
Great, thank you very much. So let me share my screen first. Can you see my screen?
Yes.
Yes, okay, great. So, well, first, thank you, Maria, for bringing us all together. And I'm very excited to be here and share with you some of the work that we've done. And I think it's a wonderful idea to try to bridge theoretical and experimental biology to understand human development, and certainly trying to build a virtual human embryo is going to be very valuable to better understand the first steps of human development. So I'm just going to share with you a little bit of what we are interested in. So of course, we are interested in this question of how a single cell can give rise to an entire human being made of thousands of single cells. And we focus on the human, and specifically on the first 2 weeks of human development. First, on the first week, going from zygote to formation of an early blastocyst, and then late blastocyst just before implantation. We're interested in the first cell-fate decisions, where the first lineage is specified, the outer trophectoderms that will become the trophoblast and then give rise to the placenta. We are interested in the inner cell mass that undergoes a second cell-fate decision, giving rise to the naive pluripotent epiblast and the hypoblast or primitive endoderm. And we are also interested in what happens after implantation, and there are a number of additional extra-embryonic lineages that are specified, that are important for the embryo to grow, but very little is understood, especially in human, as I will tell you a little bit more later. So we are primarily interested in cell-fate specification, but also increasingly in morphogenesis and cavity formation, thanks to a new 3D models of the early human embryo. So the big questions that we are interested in are: how do gene regulatory programs mediate early human development? How is human pluripotency regulated? How is cell-fate specified and then restricted by transcription factors and also by chromatin regulators? And finally, also, how is gene dosage regulated? And there we focus exclusively, well, almost exclusively on X chromosome inactivation. So we tackle these questions, and we really have made the switch from animal models to humans over the past few years. And today I'm going to share with you just 3 stories. I'm going to be very, very brief. The first story I will tell you about is about chromatin and cell-fate induction. And I will tell you, we have identified a barrier to cell-fate induction that acts early in human development. And then in the second part, I want to tell you about gene regulatory networks in human embryos. And this is perhaps the place where I'm most excited, where we could bring experimental and theoretical approaches. And then if time permits, I will tell you a bit about modeling what happens after the human embryo implants. So if we look at the human blastocyst just before it implants into the uterus, it's made of these 3 key lineages: the trophectoderm, naive epiblast, and primitive endoderm. And there is one big difference with the mouse embryo. The mouse blastocyst has the three cell lineages fully restricted before the embryo implants, but that's not the case in human. In humans, cell fate restriction is not acquired yet at this stage. Trophectoderm cells can still become epiblast cells, and vice versa. Epiblast cells can be isolated in vitro, and they still maintain the ability to give rise to the trophectoderm. So there is much more plasticity in the early human embryo compared to the mouse. It's of course possible to derive different stem cell types from the embryo, and these different types of pluripotent stem cells. Naive human pluripotent stem cells can be derived, and they resemble the pre-implantation epiblast, and then prime or conventional pluripotent stem cells can also be derived from embryos or by reprogramming, and these resemble the post-implantation epiblast. So it represents about the day 14 human embryo epiblast. And then there are different types of pluripotent stem cells in between, of formative pluripotent stem cells that have been derived as well. We can also derive other types of stem cells, trophoblast stem cells from the placenta, and also primitive endoderm stem cells. And these stem cells, self-renew in addition. They are extremely exciting tools to model development in vitro. And in particular, in the case of naive human pluripotent stem cells, they can self-renew and they maintain complete lineage potential, meaning that naive human pluripotent stem cells can be differentiated into prime pluripotent stem cells and give rise to all cells of the human body. But they can also give rise to the extraembryonic primitive endoderm and the extraembryonic trophoblast lineage. Essentially, naive cells can make all cellular lineages of the human embryo, and remarkably, they can even self-organize in 3D into structures that resemble the human embryo, the human blastocyst, and these are called human blastoids that resemble the embryo in that they recapitulate a lineage specification, at least to some extent, and also morphogenesis. They form a cavity. They can initiate an implantation response and model aspects of early post-implantation development. So these are extremely exciting models, stem cell models, to try to better study and understand what happens in early human development.
Can I ask a question, sir?
Absolutely.
So this mouse versus human, which you mentioned early on, I've heard this a few times. I was curious, do you think there's a functional, I don't know if that's the right word to use, in the sense, do you think there's some functional reason for it or functional benefit of this plasticity? Or is it just so happens? Yeah, and anything you could say a little more on?
So what comes to my mind is that it could be, so we don't know for sure why that is, but it could be related to the speed of development, and that the mouse embryo develops very quickly. So it all takes, it's much faster in the mouse. It takes about twice longer in the human. So that could be one of the reasons that the mouse embryo quickly needs to segregate, not only segregate its lineages, but restrict them to implants. And then so that's one of the possible reasons that there is might be an evolutionary advantage to faster cell lineage restriction to accommodate with faster development and the production of larger progeny.
I see.
But
does this plasticity also go to like injury? As human embryos more resilient?
So that I don't know, but sometimes my colleagues working on the brain report similar things in the brain in terms of cellular plasticity and lower restriction in the human system compared to the mouse. But it's not always clear cut or tested systematically. But I think in the blastocyst, it's really been tested very well. And so there we have clear evidence that restriction happens sooner in the mouse compared to the human.
Thank you.
So because these naive pluripotent stem cells essentially can make all cells, it was thought that they are devoid of epigenetic restriction. And so the question we ask is, are human naive pluripotent stem cells epigenetically unrestricted? And I'm going to tell you it's not the case, that even naive human pluripotent stem cells that can make everything, even these cells use mechanisms to oppose induction of alternative cell fates. So the way we tackle this question is together with Peter Ragan, Hendrik Marx, Martin Danneens, and others, we measured the proteins that are bound to chromatin in naive and primed pluripotent stem cells, and using mass spectrometry. And we also measured the abundance of histone modifications, as well as transcriptome and developmental potential of naive pluripotent stem cells. I'm not going to show you all the data. I'm just going to give you the summary. Is we found that the chromatin proteome changes between pluripotent stem cell states, and we found a large increase in several, but one histone modification in particular, the H3 lysine 27 chromatylation mark in naive cells. And so H3K27 chromatylation is basically deposited by a conserved developmental regulator called the polycomb repressive complex 2. And what we found is that if we knew that naive human pluripotent stem cells can be converted to acquire the trophoblast fate, but when we inhibited PRC2, the polycomb repressive complex 2, and then induced the trophoblast fate, we found that PRC2 inhibition improves trophoblast fate induction, arguing that polycomb helps to oppose induction of the trophoblast fate. And we could also do this experiment in blastoids together with Nicolas Rivron. And there also, when we inhibit PRC2, we have not only an increase in trophectoderm fate induction, but also an increase in morphogenesis and cavity formation in this case. So altogether, we conclude that polycomb opposes induction of alternative cell fates. So we have comprehensively determined the proteins bound to chromatin in different pluripotent states. We found that H3K27 chromatylation is much higher in naive cells, and then we found that polycomb opposes induction of the trophoblast fate. And so we knew polycomb is a developmental regulator, but we didn't know that it acts this early in human development. So I then want to tell you a little bit about another piece of work, which concerns gene regulatory networks in human embryos. So the prime motivation for this work is to understand aneuploidies, but I'm only going to talk about gene regulatory networks today. So there are a lot of aneuploidies present in human embryos, unlike in mouse, and this is thought to limit embryo growth and thought to perhaps represent a problem in the clinic for IVF patients, but not necessarily, aneuploidies are not necessarily incompatible with a human life. So to better understand the relationship between the genome and the transcriptome, we applied together with Thierry Voet a technique called single-cell genome plus transcriptome in human embryos, where we can comprehensively measure both the transcriptome and the genome of single human embryo cells. So the way it works is we take human embryos at all stages of human pre-implantation development, isolate single cells, and then separate the RNA on one hand, the DNA on the other, do whole genome whole genome sequencing for the DNA, and then SMART-seq2 single RNA-seq for the RNA, and then we can reconstruct a gene regulatory network, look at gene expression, and then do integrative analysis. So I don't have time to tell you anything about the integrative analysis or the genome, but everything, if you're interested, everything is included in the bioRxiv preprint, but I'm gonna spend some time telling you about the gene regulatory network part. So looking at the single-cell RNA-seq data, first annotate cell types. So looking here at a UMAP, which shows the cells of the early embryo, progressing from the prezygotic genome activation stage to the premorula, morula, and then trophectoderm state, and then formation of the inner cell mass, epiblast, and primitive endoderm. And then what we wanted to do is reconstruct the gene regulatory network. So we used a tool called CNIC, that was created by our colleague Steenarts here in Leuven. And so the way it works is in 2 steps. In the first step, we reconstruct a general gene regulatory network from single-cell RNA-seq data. We take single-cell RNA-seq data and identify the genes that are expressed. So we only keep the genes that are expressed, and then we select regulatory elements around the transcriptional start sites of expressed genes, and then scan these regions for transcription factor binding motifs, and then list all potential motifs in the regulatory regions of expressed genes. And so we create lists of genes for each transcription factor. We create list of inferred target genes. That's called a regulon. So regulon is all the inferred possible target genes. And then we build a general gene regulatory network with all the possible interactions. We have a big network. And then in the second step, we can measure which connections of the network are active in different cell types. So we can identify active connections by looking in each single cell at which transcription factors are expressed that target genes that are also expressed. So we measure basically regulon activity. So I'm going to tell you a bit more about that. But before that, I want to show you what the network actually looks like. So in the premorula stage, here's an example of the network at the premorula stage. And what I'm going to show you is how this network evolves from one stage to the other. And the point that I'm going to make is that what you can see is that there is dynamic change in network connectivity during cell-fate transition in human pre-implantation embryos. So it seems that in terms of gene regulatory logic, distinct transcription factors become activated and repressed at different times during early human development. So I like to think that this network data could be a nice starting point for a theoretical biologist to model something, but this is something we can talk more about. Now, what we can also do is we can plot the activity or the inferred activity of every transcription factor here in the y-axis per cell type, organized by pseudotime or by development progression. And I really love this heat map because what it shows in yellow is active transcription factors, and what is clearly apparent is that there are groups of transcription factors that are active at different times during early human development. And I like to think that this is the regulatory logic underlying the first week of human life. So what's really nice is that many of the known regulators are picked up in this analysis, but there are, of course, many new regulators that are also picked up. And so we can see nice transition from premorula to morula to early blastocyst, and an acquisition of an inner cell mass fate, and then consolidation into an epiblast fate. And then this epiblast network or group of transcription factor becomes completely different from the trophectoderm cells that acquire a completely different set of transcription factors that then undergoes a maturation phase. And then primitive endoderm cells also acquire different transcription factors. We can also use this to predict which transcription factors are most active by ranking regulon activity in distinct cell lineages. And here we identify transcription factors that have been shown to be important in mouse embryos, but also in human embryos. For example, OCT4 or PU5F1 is here somewhere. Yeah, here in the early blastocyst. And this is one of the very few transcription factors that has been knocked out in the human embryo. We also pick up some species specificity. For example, SOX2 is highly active in the morula. This is a human thing. In the mouse, SOX2 is expressed only later in the inner cell mass. So the species-specific timing of regulatory activity of transcription factor is also picked up. And then there are lots of predictions that need to be validated, and that I like to think that there are transcription factors that are important, but for which we still need to do work to address them functionally. So I told you we also, so this is it for the transcriptome and regulatory network part. We also have the DNA part, and we can identify abnormalities. So I cannot, for sake of time, show you all the data, but I'm just going to summarize what we found. So we can identify from the DNA sequencing chromosomal abnormalities. We identify aneuploidies at all pre-implantation stages. We see that, again, when a chromosome is gained, so if there is an extra copy, there is increased gene expression in cis, but we also see change in transcription factor dosage that can lead to regulatory target perturbation in trans. And we also found that aneuploidies can induce an unfit phenotype and a developmental delay that may induce cell competition and could be at the basis of growth retardation in aneuploid and mosaic embryos, and could also help explain why some mosaic embryos made of both normal and abnormal cells can, in some cases, give rise to healthy pregnancies. So I'm going to stop here. All this information is on bioRxiv, and maybe I reached my time. Or do I still have a few minutes? Or
You are welcome to keep going.
okay,
So-
okay. All right, thank you. You stop me if I go too long. So in the last part, I just want to quickly tell you about post-implantation human development. So when it comes to understanding the post-implantation human embryo, it's extremely challenging because the embryo is completely inaccessible. We really, for technical and ethical reasons, we really can't go and access the human embryo. The development is not fully conserved within vertebrates. And for example, even just in terms of morphology and morphogenesis, it's a completely different, or very different, between human or primates and mouse. It's very poorly characterized, and the models we currently have do not fully recapitulate the cellular diversity and morphogenesis of the embryo. So how can we learn more about the embryo? I'm just going to give you one small example today. And initially, we converted the naive human pluripotent stem cells to the trophoblast lineage, as I talked about earlier on. And we were indeed obtaining trophoblast stem cells, but we also obtained another unknown cell type. And so by doing single-cell RNA-seq and then alignment to a human embryo atlas, we found that these cells correspond to the extra-embryonic mesoderm. So the cells that we were making in vitro expressed all the extra-embryonic mesoderm genes and were distinct from all the other possible cell types present in the early human embryo. So what is the human extra-embryonic mesoderm? So we didn't know anything about it when we started this project, but it turns out that is a really key cell type in the early human embryo for which almost nothing is known. So what we do know is that the cells arise in the human embryo about 3 days after the human embryo implants. And if we look at the day 14 human embryo, this is what we see. We have here in blue the epiblast, and light blue the amnion, this is the amniotic cavity, in yellow the primitive endoderm. And around here, we have the cytotrophoblast in orange and syncytiotrophoblast making contact with the maternal tissue. And all these green cells here are the extra-embryonic mesoderm. So essentially, the extra-embryonic mesoderm are the first fibroblasts of human life, and they connect the embryo to the placenta, and they fulfill many important functions, such as secreting the extracellular matrix that the embryo needs for morphogenesis, but also goes on, extra-embryonic mesoderm cells also go on to form the primitive blood in the embryo. And what is extremely interesting is that there is a species-specific timing of specification of these cells. In the mouse, the extra-embryonic mesoderm appears after gastrulation from the primitive strate, from the epiblast, but in human, the cells appear before gastrulation and from completely unknown origin. So it's been proposed they come from the epiblast, the trophoblast, the primitive endoderm, but we really don't know, and there was no in vitro model for this cell type. So, and so here's what the cells look like in green in the monkey embryo. and we could confirm that the key markers of the extrahemolytic mesoderm are expressed in vitro, in the cells making in vitro, such as pimentin, but also GATA3, GATA6, posteriorin, decorin, and LUM. We confirm that many characteristics or hallmarks of the extrahemolytic mesoderm are recapitulated in vitro. And again, we can predict key regulators of cell identity using network inference approaches. So I just want to summarize what I told you. We have a new model for a human cell type that we think is very important, but for which almost nothing is known. We think this is significant because the cells appear in the embryo at a time when many miscarriages and pregnancy losses take place. And this represents a special opportunity to better characterize the cellular and molecular features of this cell type and model the primate-specific developmental mechanism. So I want to summarize what I've told you today. In the first part, I told you that we identified the chromatin proteome of naive and prime pluripotent stem cells, and we found that naive cells have barriers that oppose alternative cell-pit induction. In the second part, I told you about single-cell multiome analysis of human embryos, and that we reconstructed the gene regulating networks of the embryo and studied their impact, the impact of aneuploidies on the embryo. And in the third part, I told you that human naive pluripotent stem cells can model extrahemolytic mesoderm specification. So I want to stop here. Thank everyone involved in this work, especially all the outstanding postdoc and students, and the collaborators and the funder. So thank you.
Thank you, Vincent. So if it's okay with everyone, I will take on the theory perspective, and then we can open discussions to see how these two sides approach. Okay, so I will start sharing.
Can I ask a question while you're...
Yes, please, go ahead.
Vincent, I had a question about the last part, right? You said that the last part was hard to study because, of course, you know, so I missed, how did you guys study? Is it sort of, this is reminding me of these artificial womb type papers which came out, you know, 2017 or something like that, or maybe 15, 17, where they were doing. Is that the kind of things you guys are doing, or I missed?
Yeah, this is the kind of things that we're doing, but what I talked about today, it's in 2D still, so it's directed differentiation. So you take the cells and you direct them to differentiate into a target cell type, and what we saw is we can make more cells than what we thought. So we are learning how to make the building blocks. All the cell types that are present in the early post-implantation embryo. The next step, I think, is absolutely what you said, is to do it in 3D and to coax stem cells into recapitulating the morphology of the human embryo in 3D at post-implantation stages. And I think this is where the field is going to go.
I see. Okay, cool. Thank you.
Well, thank you everyone for joining. I hope you can see my slides. Yes.
Yes.
Yes. Okay. So thank you, Vincent, for the great introduction. I'm actually going to go right through my question. I think you showed a single cell that went to a baby, and we have the same question. We're wondering how does a single cell generate all the diversity that you see in an organism? And we know that we start from a single cell and that undergoes some sort of state transition. And we can characterize these different cells by morphology, some sort of developmental plasticity, and even by gene expression. So most of us look at a cell via genes and try to see whether we can distinguish one cell type from another by looking at their genes. So for me, it's how does a single cell know exactly when to change, and how much of these cell types do we need in an organism? So we are looking at what affects the cell behavior from an intrinsic factors, such as ploidy, cell cycle duration. You can have a different genome active state. Different genes can interact within that single cell and affect how it's going to react to its environment. Specifically, there's also gene length, which I'm going to be talking about a lot more today. transcription rate, how fast these genes are transcribed, and how fast are they degraded. Those can all affect the cells from an intrinsic perspective. So the way we do this is by using mathematical modeling to simulate the different developmental systems. And we try to integrate different knowledge from various disciplines, which includes the mathematics, physics. I also include evolutionary game theory and geometry. And these types of models provide a way to explore these factors individually. So we can combine one or two factors together or eliminate some and see how they affect the overall system. So just to also say that this platforms works in 3D space and time, and you can do this all at a single cell level, and then we can export the data in a matrix fashion where you can analyze the system the same way you do single cell on regular experiment. So it just gives us a way to look at and compare experiments versus modeling predictions. So what's unique or different from other models is that I incorporate geometry. Unfortunately, I'm not a mathematician, but one of the ways I was able to, access mathematics and learn really quickly during my PhD is using geometry. And so I started in a 2D system. I modeled sea urchin skeletons during my PhD. And when I joined Gary in 2016, we worked together to create it into 3D. 3D geometry is another world. And we create algorithms that try to mimic how cells may coalesce, like they work together. So there's lots of theory that showed that cells can go in a fluid to a solid state. And so the model works with geometry to try to mimic this behavior. Oops. And so this allows us to basically track cells across time and see how they grow and interact. And this could change the shape by just changing how they polarize or affect one another. But as I was building this model, I started asking just simple questions: how fast do cells divide in early development? It is known that they start really fast early on and then they slow down. But it wasn't clear how fast is fast and when do they slow down? Is it just an abrupt change? And it was then, well, at least I realized really quickly there's some gaps in our system. What does cell division do? So we started collecting a lot of data. Here I'm just showing you for the mouse, but we collected it for as many species as we can. And I start to see that cell cycle duration, it does change from slow to fast, or from fast to slow in early development, but then it's different for different species. But what was more important, it was different for different tissues. So why would you have different cell cycles for different tissues? So our first paper, which I'm not going to go too much into details about, but I need to tell you a little bit to set the stage. What we did is we modeled a simple cell that carried a genome, but this genome can have genes of different length, because the idea was, what if this fast cycling state is actually not just creating a lot of cells with just a specific cell number, but it's also limiting the type of genes that could be expressed in a system? Because what if we had longer genes, and if these longer genes need more time, they're not going to be seen in these early fast cell cycle durations. So the model assumes that we have genes of different length, and shorter genes are going to make more copies because they're going to be able to be transcribed faster than a longer gene system. And by just exploring this simple model, we were able to see that there's sort of a trade-off. Early cell cycle durations that are running really fast are generating these fast numbers, but they come at a cost of diversity. They're not diverse. They will look similar to one another, but as the cell cycle duration slows down, it can introduce new genes into the system, and that will generate this diversity. So I hope I'm not going too fast here, but I'm going to show it in one more figure. So if we assume development time is going downwards, if our system carries two genes and they're both of the same length, so X and Y, and they both carry the same length, LX and LY are the same. If you start dividing at a fast pace, we're going to generate just one copy of an XY, and we may just have three cell states. So X, XY, and Y. And those are just shown here in pink, orange, and yellow. Now, this sort of, platform or sort of a transition. If you transition from fast cycles to slow cell cycles, and we allow it to happen at this time point in development, all of a sudden we're, let's say, able to create 2 copies. Maybe I double the cell cycle duration. And right away, you can see that there's more diversity into the system, because now we have different combinations of cells that can be generated. In B, all we did was change the time of when this transition can happen. So if you go from fast to slow really quickly, you don't see these early fast cell cycles, and right away you explode in diversity. Now, how does gene length play a role? Gene length all of a sudden comes in. So if X and Y are not created in the same abundance, and if the cell cycle duration is cycling too fast for Y to even be completely transcribed, all of a sudden you're going to see a whole state of just cells that carry gene X. And as we transition from fast to slow, it is then that you'll see Y being introduced in the system, because now there's enough time to create Y. But what it also affects is the population proportions. So by just changing the length of Y, we're able to also change how much of these types of cells that you can also create. So cell cycle duration and gene length can influence diversity and the timing of when you see certain genes. So we went on right now to talk about, to then explore, okay, so we know that cell cycle duration seems to influence the system, and genes and their gene length seem to influence the system, but there's a whole systems biology, a whole field of work that has been done on gene regulatory networks. Genes don't just float around in a cell, they actually affect one another. And so here I'm just showing you, basically our first paper, our first work just covers like a little portion of our understanding of what genes do in a cell. So we try to explore what happens if these genes interact, but also have different gene lengths. Because to date, nobody wondered why genes, whether genes would have different length, and how does it affect the timing of a system. Because what we know is that if a gene is longer, it's going to take longer to be transcribed. And that might be important for certain species, because you may not want to see a gene occur early on. And we see this in brain cells. A lot of neurons have longer genes because they don't need to be, they don't appear in the system till later. Muscle cells also have quite long genes. So this is, the other thing that is important is we found, and I'm not going to go into a lot of depth into this, but we also explored this for other species, that certain genes, if you group them by just length, they access certain pathways. So there seems to be, during fast cell cycles, if you can only generate certain short genes, then they're going to only access certain functions. And if you group only the long genes together, then we see that they access different types of pathways. And of course, some pathways have mixed genes of short and long. And it starts, you can beg the question, you know, how do they control development, and what part of development do you need a mixture of short and long gene? But what we want to know is, how does this gene length affect the transitions of cell states across time? If I'm going too fast, please slow me down. I just want to give you a big perspective of the theory. So this is just quickly to show you that we looked at different species, and different species have a broad genome. This means that you can have very short genes, and in humans, it can be as small as 55 base pairs and as long as 2.5 million base pairs. And so this is sort of like the breadth of the distributions of the genomes. And if here we mark, let's say everybody, all the cells are transcribing at the same rate, and we count 1 hour of transcription, you can see that there's a lot of genes that could be missed, especially in the mammalian systems. So because they carry longer genes, you're going to miss out almost 20% of the potential genes that are available to you. The other thing that, this is just a theory, but we're proposing that maybe this is also important for developmental time. So one of the questions was, well, what if I plotted all the different species that we could find and their medium gene length and with respect to developmental time? So there's not a lot of data out there, but we sort of curated as much data as we can about when a species sort of, if you laid an egg, I basically assumed it's when it was hatching. And if you're a mammal, I waited till the end of gestation period. So as soon as the baby is born. And so you can see there's, C. elegans and very small, eukaryotes tend to have narrower, let's say, genomes, but they also develop faster. And the species that take longer time to develop tend to also have these longer genes. Now, of course, I don't know what, and they might have been co-evolutionary, but this is also important. So if you're trying to understand developmental time, gene length, and cell cycle duration, there's more to it than just creating cells in numbers. Okay, so how did we model all this?
Yeah. Can I ask you something, Maria?
Yes, of course.
Very interesting data. So I wonder for the gene length, do you think that's it's like because it's the median that changes. So do you think it's all genes can become longer if development is slower, for example, or do you think that it's just a few genes that become very long and that's driving the median higher? So do you see what I mean? Is
Yeah.
this driven by all genes or a subset of specific genes that become
So
longer?
not all genes changed length, at least when we did like sort of an exploration. So when I mean gene length, I probably should explain it's exons plus introns together. And there's a whole hypothesis across evolutionary time. Species that needed, let's say, genes to show up later, they had new introns added. And when I looked at the genes, the really long genes have almost like 1 million base pairs of introns, right? So it's almost like the protein size did not change, but the gene length, how long it is to transcribe, had changed. And when we looked at it across species, there are definitely certain genes that increased in length across all these species. There's distinct ones and ones that didn't. And I read a paper that once proposed that it was all the new genes did not have any introns in them because they haven't had time to evolve and change their timing. So it seems like the newer the genes is in across the evolutionary time, the less introns it has in it. So if a human, if I were to guess, I would say human specific genes that only evolved during human time may be shorter than the ones that, let's say, evolved in across species, across developmental evolutionary time. So now we have different times, right? Evolutionary time is something and developmental time is something else. And so the genome and all that was sort of like, there was a different selection of it. And now development is using it or somehow it's a co-evolved system, right?
Thank you.
You're welcome.
Okay. Maria, may I ask? The question that I just joined, and I find it very curious and tempting that for the first time I see connection between length scale and time scale, and it's happening in the cell, which means that there could be another mathematics here.
Do you mean developmental time? Is that-
No, time, cell cycle, time, and what you say, developmental and evolutionary, it's all about time.
Right, there is
Now
different
we
time
talk
scales.
about gene length.
Right,
So there is something called scale separation and also scale similarity. And this is somehow, in a funny way, also connected to spatial temporal analysis, but that's a little bit more remote. I'm talking about some basic mathematical analysis between those scales.
right. And it's actually really hard to attach all these scales. I have, Gary has a PhD student who's helping me write some sort of equations to see if we can connect these different time scales, but it's actually really hard. But how about we move this to like discussion, because I want to like run this another 4 minutes. But Amy, you have a question?
Yeah, and I think maybe you will talk about later or not, but I was just curious, have you looked at whether there was any sex-specific differences in terms of gene length for certain genes, and also whether environmental factors might play a role? So if you've looked at these same genes across different, let's say, cultural groups or ethnic groups.
I don't have data or know of data that, separates these two things, but it would be interesting to see, let's say, sex-specific cell types, and then we can look at the various genes that are expressed, and then we can see if there's a gene length difference. I mean, right now, I assume the data that we get from a single cell data, let's say mouth, cortex, I just take the whole system and I separate the system based on cell types that everybody checks. But another way we could do it is also separate it based on sexes and see if there is a difference between them, but I haven't. That's interesting. Okay, well, I'm going to get to the more recent and at least the fun elements. I mean, at least for me, gene lengths seem to say when, if I want a certain cell type to appear in a system, and I mean, even Vincent indicated there's a difference between how fast certain species develop, and there's a difference between mouse and human. And so maybe this gene length can tell us when something occurs, but also in just developmental time, we're always looking at, here, if you look at this, we always look at time across divisions or some sort of states, and then we always see expression, but maybe this gene length can show us when we should expect a certain genes to appear in the system. And so the way we model this is we start with a single cell, but every cell has a genome, and this genome can have different gene length. And now I'm adding a gene regulatory network to the system. And since this is theory, we're going to explore as many gene regulatory networks as we can. So we're going to go from a 2-cell state to the 3-cell state, but I'm going to keep it very simple for just the talk. And so here, what I'll also show you is, let's say our first theory was just X and Y, but they don't interact. And across down, for each row, we're going to change the gene length of these systems. And across each column, I'm just going to show you 2 examples of different gene regulatory networks. Let's say X activates Y or X suppress Y. And I hope you can appreciate that even just for the first column, where if you assume X and Y to be the same, we're going to expect that they're going to be present in the population at the same frequency because they're the same. They're going to be expressed the same amount. And so you're going to divide them. And so you might have 2 equal cell state. But as soon as you change the gene length, so X is smaller or shorter, and so there's going to be a lot more X in the system, we change the cells that are potentially available for us in this dish or in this population. And if you make, in this case, X longer, now there's going to be less X. And so you're going to have a different cell type by just changing the length. And now if you change the interaction, if you look at the last column, you can also change the timing of when you see a certain cell take over. So we know X is going to suppress Y. And if they're the same length, then they're going to compete, and X is going to take over pretty fast. And it's going to even be faster if it's shorter. So this would be the second row. But if it's longer, then all of a sudden we're going to have this state of Y being around a little earlier, and then X taking over and suppressing Y. And this could be something that happens in development. At least from when I read the developmental books, you see that gene X and Y are both available, and X will end up suppressing this other gene in time. And there's always these repressors as opposed to these activators. Somehow there's a lot more repressors, and this could be one reason, right? This is just another potential. So we ran a lot of simulations, but I just kind of want to show you what happened now. What if we change cell cycle duration across these rows? So you can have an increasing cell cycle duration, a constant cell cycle duration, or a decreasing cell cycle duration. Again, I'm just showing you a different gene regulatory network, but just so you can see what happens. In the columns are gene regulatory networks. Time is on the axis. In this case, X is larger than Y. And just looking across the row for the last column, you'll see that you have a completely different dynamics. And all we did was change the cell cycle duration. So you can go from a system where you basically only see Y in the cells versus a system that X takes over, and now you have a completely new cell state, versus one that's going to fluctuate between 2 cell states across time. So you may see one go up and down, and we do see this happening. And this is just caused by changing the cell cycle duration. Okay, so again, this is fast, but I just kind of want to get you through the theory. So before I jump into the example, so I showed you the first A, B, and C, where we have when we can change the cell cycle duration and time and gene length. But now if you add a regulatory network to the system, so let's say here Y suppresses X, and X is smaller. So this is just, you can compare C and D. All of a sudden, we're going to bias the system to more cells that have a a Y type as soon as the cell cycle duration slows down, but you may also eliminate other cells or remove a whole set of systems in time. So then we're going to see a system that has only X early on, and then, you know, some other transition states that kind of explode and then bias the system to one lineage. And what I think is happening in development is that different tissue is running on different cell cycles. And then you might see them going down and branching out in across states, but that's just a theory. Okay, so this is kind of the fun part. Does this work? Does this apply? What can we use this for? And I'm only going to talk about one particular, you know, application was trying to understand the transitions of the 3 germ layers in early gas relations. So there are, this is a big transition, so it's not a simple things, but there are 3 types of cells that emerge early on, and they're called the ectoderm, mesoderm, and endoderm, and they can generate all the different types in your body. But I'm only going to talk about this early first steps. The other thing that's important is that you could potentially distinguish these types of cells based on their genes. And so the endoderm would have SOX2 and some other, and so it's important to distinguish them based on genes, because of course that way I can apply the gene length. And the system that I'm going to try apply is part of these micro patterns that they use single cells, but human stem cells, and they can apply them on these little tiny dishes. So I don't know too much. If anybody here is an expert, you're welcome to intervene. But what's interesting is they see these plates with a certain pattern, and then you can see different cell types emerge across time. And Peter Zanstra and his group do this. And so in the center, you're going to see one cell type that mostly has SOX2 in it. And then as you go outwards, you're going to have the mesoderm and then the endoderm layer, and I think the trophectoderm. And each one of them is carrying a specific cell type. So how do we do this in our model? Okay, I'm going to use the exact same model that I did these toy, but now instead of having this 3 or 2 gene regulatory network, I'm going to add this little small hairball. And I know it looks very small, but it took me 4.5 months to put it together. And this is done just paper-based. So we took as many papers as we find, as much, and it was hard to find things just for human because there are differences between human and mouse, and certain things that they find in mouse do not apply to humans, especially let's say between OCT4 and CDX2. They don't interact the same way as they do in mouse. So we tried to do a good paper-based GRN. Vincent showed you how he's doing one based on actual experimental data. Maybe somehow we could merge and see how that would work together. The other thing that's important for our model is to show the gene length. Each one of these genes also carry gene length, and they vary. One thing to note is SOX2 and the ones that are all famous for stem cells tend to be shorter. And as you differentiate, some of them become longer. And the other thing that I'm not going to talk a lot about, but BMP4 is more like a morphogen. And there's a lot of evidence that it also affects cell cycle duration. So I'm assuming that is happening in the system. And if that is true, then what I could do is run simulations where we know nothing about cell cycle duration in the system, but what I can do, I use that as my parameters. So for the same gene regulatory network, for the same genome, all I do is change the cell cycle duration, and you see 2 different cell types. For really short cell cycle duration, we're only going to see SOX2 in the system. If I increase it at some sort of rate, all of a sudden you see the crazy interactions happening. Now, we don't know if all of this is true to the system, but what I can do is change the cell cycle duration. I can do it as many rates as we want. And then what I can do is show what's happening in this population across time. So early on, irrespective of what I do to the cell cycle duration, you see that early fast cell cycle durations tend to show more SOX2 and OCT4. And I just want to show that in micropatterns, at least in the papers that I read, nobody really talks about what's happening across time, although like I know I twisted here, but the experiments measure things in time, but really what they're looking at it in space. Unfortunately, I'm not doing things in space, but somehow the cell cycle duration changing in time seems to reflect what's going on and changing in space. Another theory that we had in the first paper is maybe that cell cycle duration can help you create boundaries. Like if if different boundaries or different regions are cycling at different rates, then maybe then you can create these different zones. So that could be something what's happening here. But what I find is quite interesting, it's almost mimicking what you would expect in this system. So we move from lots of SOX2, which would be what they find in the center. So that could be the fast cyclers. And then you go to this other system with a lot more CDX2 and TB and T. I'm not saying this is like capturing exactly what's happening in the cells, because we want to know, we don't know what the cell cycle duration, but we know that by changing the cell cycle duration, we're accessing something that seems to be close. But, and this is the fun part, is that it seems to be close. So these are I just copied and pasted from 2 different papers that study these micro patterns. And this is the dynamics that they see is happening in time. And so we see very similar. We see SOX2 going up early on, and then later you have the changes between TBXT and CDX2, and it seems to be quite close. So somehow by just adding gene length into the system, putting a regulatory network, and I want to also say that there are no threshold. These are not switching at any threshold. This is actually, I think people who model will understand what I mean. Usually you have threshold that switches between one cell state or another. I'm not doing that. There is no threshold, there's nothing, and you can see that somehow the gene length seems to capture the timing of these transitions on its own. And that is pretty cool. I hope that. So the story is that if you assume gene length could be a true biological parameter that we can now apply to our gene regulatory networks, we can start to change cell cycle duration to see how it might influence certain portions of our developmental system. And here I'm just showing that if you change cell cycle duration, which is across the rows, you can change the dynamics of the system and what you see appear and disappear. And if you assume that it's equal to gene length, because I'm sure everybody's wondering if that makes a difference, it does change the dynamics. So by just adding the real gene length that we have access to in the databases, it seems to capture the timing of these events. And for me, this is, I mean, it's a big step. There's so many mathematics and physics equations, ODEs and PDEs everybody talks about, but now all of a sudden we have a biological parameter that maybe we can grasp and use in our systems and to help us. And this may have not been important for, let's say, the systems that use bacteria, because a lot of them had very similar genome length, a lot of the bacterial system. But as soon as we transition into mammalian systems, now it becomes quite important, because timing of when these cell states occur is important. So I'm going to stop here, but I also want to show you that we tested it on the neural tube with a little tiny gene regulatory networks, and we can also see the transitions across certain regions, which is quite exciting because we can apply it. But the important part is to actually get the regulatory network that is true to the system, which is an important part. So I want to thank you all for listening to me, and of course, Gary, Ruth, and Brendan, who helped me with all the work in the first paper, Andres, Esther, Janet, Nika, Derek, and Peter for supporting and helping this work, because they lent an ear and advice and a lot of important information. Of course, I don't want to leave this by not thanking the Stem Cell Network, because of them, we're able to join and talk today. So thank you all. I hope I made it in time. Let's stop share. And if you have questions, please. Sorry, we're a little over.
Can I ask a question, Marie?
Please, go ahead.
So just wanted to understand the model. So the idea is that everything resets at cell cycle time. So in the sense, when the cell cycle starts, then you start expressing the genes, right? And the regulatory network, so if as you had Y suppressing X, right? So if Y is too long, it's never made in the cell, and so it cannot suppress X. And the next time the cell cycle comes again, you again are Y is at 0. In the sense, there's no inheritance, right? If you could imagine that they could just inherit the state, then this wouldn't matter, right? Or am I missing something? Or
No, there's actually multiple levels. I mean, I'm
Okay.
hoping to show you that this is sort of like a default state. At least in these early developmental system, they tend to be more loose. The chromatin is more accessible. And of course, as you transition to different differentiated states, you're not going to be as accessible. So you need to activate. But there's evidence that if you are a long gene, and this was done in the fruit fly, so I think the UBX gene was really long. And what they tested is if it didn't get completed by the end of the cell cycle duration, it was degraded. And then you started from scratch. But another thing that's cool, I think everybody hears about maternal transcript. It's not as important in humans as it is in other species, but early on in the early embryos, at least in xenopus and zebrafish and some mouse, there's a certain transition between the maternal state until the cell cycle duration can get longer. There are maternal transcript that already exist in the cell. And when they analyzed these transcripts, they found that they're much longer than the early zygotic ones when the transition happens. So I would say like it's almost another mechanism that you can superimpose. Like if you know that the cell cycle duration early on is going to be short, maybe some evolutionary pressure ensured that the longer transcripts that you need early on that won't be transcribed are sort of incorporated into the system. So there's many layers, but here I'm trying to, because you need to unravel all of these, we're trying to assume the basic null state. Later, we should add like degradation rates. Maybe that, you know, if you get to stay in the system longer. And I do run simulations where you have the probability of being removed versus probability of staying. And that
so
will affect.
in your currently, basically, you take it to 0. You allow
Right.
it to,
This
right?
what I'm showing you here,
Right.
because, you know, first we want to figure out what happens in this interaction, right? I think Vincent showed a nice picture where he shows, you know, for the same interaction network across time, you're going to see different genes that are there versus not. And our theory is kind of showing you, well, the gene has to be present for it to create this interaction. And so now we can sort of show maybe why certain parts of the networks are accessible at one point versus another, even though the whole system is supposed to be there. But because the genes are not there, you're not going to see these being active. But of course, we have to disentangle these.
Maria, Aditya
Well,
here. I had a
wait,
similar similar line. So when you are saying about gene length, it's just a transcription you are considering or translation as well?
just
Because
transcription. This is just
only
transcription.
okay. And
Because
while
I was going to say, I'm only going up to what I know, and this is single cell RNA-seq data. And so right now I have only knowledge up to the transcription level.
okay. And while cell division, there is a probability of symmetric and asymmetric divisions, because of which there is a difference in cell types as well. Are you accounting for any of those in your models?
So that's another like loaded question, right? So just defining what you mean by asymmetric and symmetric division. Do you just mean that they're cycling at different rates? Do they carry
They are
different genes in the
they are
system?
dividing at the same time, but the point of number of transcripts which are going in one cell versus other cell is not equal. They are
Right.
like different.
So
So
we
instead of
assume random distribution of these transcript. And so if you do see a single long gene in the system, all of a sudden you're going to create asymmetry on its own, right? All of a sudden these transcript counts and how they're going to be randomly divided into the system so I just assumed that because I don't know any other knowledge. So I assumed random distribution of these transcripts. But if you have one copy of long gene, then we're going to just have one cell that carried this long gene and the other one that doesn't. And that's going to be affected differently than the one that doesn't have these long genes. And then on its own, the simulation is going to turn on and off these genes across time.
okay, and this is a 2D model, right? Are you planning to implement in a 3D agent-based model where the interactions also play a role?
So I know nothing about what's happening in 3D. Like the model runs in 3D, but I just took away that option because I don't know what's going on between the cells and all that. So right now, yes, it is just a population level or single cell level, but I combine the information. But once maybe we know a little more with organoid systems and that's the hopes. I mean, first of all, just saying how cells change states was hard without having to put them in a specific. So there's a lot of way to model this or get past it. In order for multi-scale models, what they do is right away, they have a threshold, then you become a certain cell state. But I didn't want to do that right away. I wanted to say, how would we distinguish these cells without setting a threshold, and can I get some of this information? But it is quite hard. I mean, I hope it is seen. Knut, you have a question?
Yeah, thanks. This is the first time I'm attending, so I'm really happy to be joining this group. And I'm really sorry, Vincent, I missed your talk. I was only able to log in later, but this is really fascinating to me, Maria. I think I had a lot of questions that I think were somewhat answered. I mean, one of them was what length really constitutes, and I think the last person who questioned you is also asking about things that, like transcription, sorry, not transcription, but translation rates. I'm also thinking about things like splicing, how splicing is regulated, the number of introns that you deal with. But to go beyond that point, maybe I'll get to a deeper question is, have you looked at variant isoforms of genes? Because that the usage of different like splicing variations will change the length without, like, the gene itself is long, but depending on how it's expressed and how it's spliced will actually change that particular isoform for that particular cell type. So have you looked at that over like variant isoform usage?
So I know that they exist. Of course, transcript come in different length, and we see that accessible, but there's only one project right now that we have any indication that isoforms play, and let's say in the muscles. So I'm just trying to say like DMD. I'm just trying to figure out what my brain lost, but it's one of the longest. Titin, they're about 3 million base pairs long. But muscle cells, when they're in quiescence, which they are in quiescence a lot, often, they tend to have that same gene, but in a really shorter form. And so one of our proposal is maybe in order to maintain like sort of memory that you are a muscle when you quiesce, you might want to keep this short transcript, because quiescent cells tend to have a low metabolic state. They don't want to be doing too much stuff, and a lot of their activity is quite suppressed. So maybe you don't want to be transcribing a really long gene all the time, and you might want to transcribe a really short version. That's just a hypothesis. I don't know how we're going to prove it, but that is one idea, is that maybe one way to, because another thing is, how do cells know what they are? And when they regenerate, that's another thing. We were talking about development, but regeneration is another one. And how do they know, when do they start off from? Because there's a lot of evidence with regeneration. They kind of know where, like if you cut off a limb of axolotl, it kind of keeps going. And so there must be some indications you are here. You are a muscle cell when you wake up. And I'm guessing that's one element, but the truth is these mechanisms evolved to bypass this default system. And so there might be other reasons. And I think we need help to work together. I mean, I can only theorize so much, but in terms of these different sizes, this is the only evidence I have that might be one way you can use them, but I don't know what else you would. I mean, you might want to be able to differentiate between two lineages, but then we wouldn't know, because then long SOX2, short SOX2, does this mean it's a different lineage, or is this the same lineage? But right now, the way we look at them in single cell data, SOX2 is SOX2, right? We're not comparing long versus short version of it.
Okay, maybe another way to look at this, because I'm starting to think about other potential changes to gene length. One is, has anyone artificially perturbed this by artificially elongating genes with introns? Or the other thing that I'm thinking about is in more natural conditions where you have like mutations at splice donor or splice acceptor sites in disease. I mean, most of the time that results in some sort of frame shift and you get a truncated protein. But I wonder if there's some sort of natural situation that's been ever described where like exclusion of particular exons has led to a disease without resulting in a frame shift in the protein.
So that's interesting. I've never read or even looked for the exon changes in that shift, but Nika Shakiba, she couldn't be here today, and I are trying to create in mouse ESC cells some way to add or elongate a gene that doesn't affect the system and see how does that change with cell cycle duration. And she's,
Mhm.
I'm not doing it, but she's doing the experimental part where we're working together. She has somebody, and it's not that easy because as
When I
soon
was
as
in
you change
Androsh's
the
lab,
length-
myself and there was a Japanese postdoc there. I shouldn't say myself. He did most of the work, but we were working with artificial introns that we were dropping into like selection markers and things. Not for this purpose, but I think those artificial introns could be used in this case too.
So maybe we should meet because Nika's working on this and
Yeah,
she
sounds cool.
um,
Can I
her-
just throw in one more thing? I know other hands are up, but the other thing I noticed in your discussion is that like neurons, muscle, like you said, they have very long genes. I'd never thought of this before, but they do have very long genes, but they also, most of them are post-mitotic, or they have like weird cell morphologies, like muscle cells are multinucleate. They don't really divide. They'll just generate multiple nuclei. So I'd never thought about this before, but it's just fascinating to me that a cell that has a very long life without an active cell cycle correlates with very long gene length. I'm just,
That's
it's
the
kind
whole
of a
point.
statement, sorry.
I mean, it's more about a perspective, and I kind of want to show, like I said, it was more like a missed gap because everybody's like, we know everything about cell cycle duration, we know everything about gene length, you don't need to worry about that, but it didn't fit in. I couldn't just add it into the model. And so it's kind of interesting. It took us to one side, but it's definitely another perspective, almost like a default state that nobody paid attention to.
Yeah, it's
It
very
doesn't
cool.
negate all the other states, right? It just gives you another perspective of why you would have maternal transcripts or why you would add a different way to maintain the cell's state. I hope that's the whole point of this discussion. Mark, you had your question up. Oh, I can't hear you.
No, you're right. That should, even after all this time, we still forget to unmute ourselves. I guess more comments than questions, or maybe a question to start with. So I guess when you were doing your simulations, you had to convert gene length into time somehow, right? So I guess you used a mean transcription rate and some amount of time for splicing, I guess.
No, there's no splicing. I stopped before splicing. So
oh, I
this is
see.
really
Okay, because splicing, so there's lots of data that suggests that splicing is not only co-transcriptional, but it tends to, the transcription tends to slow down when you hit a splice site, and maybe even pause in some cases. And for the few cases where people have measured the expression time of a gene, you get kind of a reasonable estimate of how long that's going to take if you assume it takes about 5 minutes per splice site, and add that on.
Okay.
So dystrophin, for example, which is this massive gene, takes 16 hours to
dystrophin.
transcribe, right? Yeah, so if you kind of assume a transcription rate of 70-ish nucleotides per second and 5 minutes per splice site, you get pretty close to that 16 hours. So that's I guess partly a comment. The other thing was something that occurred to me while Knut, I guess, was asking about kind of artificially changing the number of introns. There was a really interesting experiment done a few years ago, and I'd have to dig out who did it and exactly when, but where they did the opposite. They were looking at somatogenesis, where we think there's a kind of a clock operating that generates the somates. And what they did was that they, in one of the genes involved in that, and I'm sorry, I can't remember which one, they actually removed all the introns and they made an intronless version of the gene. And all of a sudden the thing didn't work anymore. So people have, kind of tried to think about timing and how that's affected by fooling around with the number of introns. So, I mean, what you're doing is a really interesting addition to that literature.
Yeah, no, this is not new. I hope, but I think it was just the right time that I could apply a model. And I think what happened early on, like when early experimentalists saw this, and I think Edgar, I'm trying to remember, Bruce Edgar, he worked on Drosophila, and I believe that he was one of the ones who saw that there was a distinction between the cell cycle duration in these early development time points, but there was also another competition. I believe Kirshner and Newport also saw in their frog embryos that it might actually be like the cell size thing was more about the cytoplasm versus the nucleus, and that took off and very little effort. And it was really hard to actually access the cell cycle duration. And believe it or not, it's still hard to really know how each cell is cycling. Like there's different mechanisms, but it's still not a very accessible thing. But a lot of people thought about it, or at least recognized that it exists, but it was hard to prove or measure. And this model just kind of gives you another potential. Modeling can do this. I mean, I hope everybody could see that this is more of a two parallel perspective, but somehow if we joined, we could, you know, have strength, because what Vincent was able to show and what he's able to do in his experiments. I mean, when I saw his heat maps and you saw the different transitions, and each section across time had different transcripts and they changed. My first question is, I wonder if they're changing in length, right? So that
we could look
I
at
don't
that.
know, but
We could look at that.
right. I just meant like, what if there's a different, what if they're also accessing a different part? That's just another, because it was clear blocks, right? And so it would be just, it doesn't negate what we already know and what we're doing with gene expression. It just adds this another parallel perspective or characteristic that we can also look at the cell cycles. Maybe some of them are cycling faster, and we can not only distinguish them based on their single cell expression, but now we can also distinguish them based on their gene length expression, right? So that's just another character that we can add.
So I really like the model that you built, and I would be really excited to do something similar for pre-implantation. So what's the minimum amount of information that we need to know? We need to know a few core regulators and their interaction, whether they're repressive and activating. Because right now we have a big network, but we don't really know what is doing what. I think we could predict what is activating, what is repressing. If we would trim it down to core regulators, would that be enough for you to try to build something?
Yes, I mean, in the neural tube, it's down to 5
Great,
genes.
great. And pushing it even further, because in the pre-implantation human embryo, we don't know what the few core regulators are. Would it be possible to test different combinations to actually find out what the regulators are? See, what do you need to have to recapitulate what's actually happening?
Exactly, and thus I've done that actually in the HESCs. I mean, what I show you is one example, but we have like 100 tests, and you can take away some and add and see. I mean, I didn't talk about it, but a lot of the work is done on what is called Turing patterns and their self auto-regulation. What you see is the network that I showed does not have auto-regulation, and then you can test it whether you need auto-regulation or when you don't need it. If you add SOX2 and then like you could actually, the neural tube one, the network that everybody uses is not the one that I used. It's the one that I took from papers, and I am showing that the ones that people think is real is not necessary, because they use these hysteris and they're trying to explain it with some ODEs, and that might cause trouble. But like you could actually test these things. I didn't go into depth with it, but you can start to say, you know, what is the network that you need to generate this pattern that we see, and what does it mean for us?
Very cool. We should meet and talk more, and I would be excited to team up to try to build a model for pre-implantation.
Thank you. That'd be great. And thank you for joining and agreeing to talk. Serge, you have a question? We're not going to go too long. We're already half an hour over. I'm sorry, everyone. But I hope you're enjoying this.
Yeah, it's very interesting. My question was, have you placed like the number of cell cycles in context between your model and what you see in vitro? So for example, you showed that patterning, right, the micro patterning, and then in your simulations you showed that like on the time axis you had like number of cell cycles, right? So I was just wondering, like, how many cell cycles did you need to get that pattern, and does it match up with the in vitro data?
Yeah, I didn't need to have that many cell cycles, and it sort of almost, because I like my cell cycle, which I call a division, it's actually reflecting what I think is around the time that is happening in these human cells. I mean, there's a warm flash paper had like some distribution of what is the cell cycle, and I start at some point, and it's supposed to be like relative, but the thing is, it's all about relative, right? Like, so I have a transcription rate that's an average cell cycle that is potentially, but if it's sort of robust within a certain range. And the only way that I will know exactly what the cell cycle is for specific species is when we start to measure them in different cell lines, because they do differ between cell lines and what they grow on and all sorts of things. And I don't know that, but right now, what is interesting is somehow it's showing you cell cycle changes in time, seems to reflect what is we see across also space. And it's just adding another what if to the system. But you need experiments and sort of like this bridging two sides to really understand what is going on. All I'm doing right now is proposing another potential of what this could be, but I do not have.
No, I appreciate that. That's cool. I was just curious. That's all. Thanks.
Yeah, no problem.
Can I add one more point?
Sure.
So I want to go back to this discussion of like artificially changing gene length by adding or removing introns. There was a couple of papers where they were trying to minimize yeast genomes by removing intronic sequences, and yeasts just naturally don't have a lot of introns.
Right.
And they don't have a lot of like transposable elements and repeat sequences like complex mammalian cells do. So I wonder if you could apply your model to compare like some of these new yeast species that have lacking introns. I don't know if you see like accelerated cell cycle. I'm not sure if they reported that in the papers, or if, I don't know, how it changes different like gene expression networks. I think that might be an interesting place to look as well.
Like I would say like it changes the expression network, because for the yeast all of a sudden changes cell cycle duration. It might. I mean, I don't know how cell cycle duration, this is another question in itself, right? Is cell cycle duration changing because now all of a sudden a new gene was accessed and then it does something to the system and it's sort of like self-moving across time? I don't know. Or does the cell just going to have like this default state? It cannot be just one thing. I'm assuming cell cycle duration is also influenced by its own whatever's in the system. So I mean, I didn't talk about the neural tube, but there's evidence that sonic hedgehog, and they see this in brain cancer that have the sonic hedgehog, and the cells are cycling faster. So I assume this. So in the neural tube, I assume that it's going to go down faster whenever you see sonic appear. And that's how I was able to recapture what's happening in the neural tube. So I do need to know the direction of how the cell cycle duration is changing. But right now, it doesn't seem to be necessary to have the exact same one. I just need to be approximately to what it has to be biologically relevant. And I think just because biology for me is like how I lead the question. I mean, everybody has a different question. And for me, I find that's the most important part, is the fact that we're putting a model that's using biological parameters, or parameters that we can define biologically. I mean, I could have called them alpha, beta, gamma, but as a non-mathematician, for me, that was hard to access. And now I feel like, oh, well, I know what alpha is, that's cell cycle. And I know what beta is, this gene length, and I might be able to get that. And gamma might be another thing that we measure. And it's not that others didn't do it, they just called it something else, and it wasn't biologically accessible for us to, right? So that's where I think Mark here is the expert in all these dynamical systems, and he probably knows. It's just, I mean, there's a lot of parameters. And I find value that all of a sudden we can say what parameter seems to give you something that distinguishes from one cell to another, or even potentially different species. And we have access to it. So are there any other questions? Sorry, it's way over time, but I hope everybody benefited from this discussion and enjoyed. I definitely enjoyed. Thank you, Vincent, for being here.
Thank you. I very much enjoyed too. Thank you for the invitation and for organizing this great group.
Well, thank you all for joining, and have a good week. And if you have time, join us again Thursday. We have another talk. Okay, bye.
Okay, thank you. Bye.
Bye.